single person like Alicia and with all her enthusiasm to be here with us today. So she's going to be talking about her talk, which is from genetic to species diversity in the Mexican skylands. So with that, I turn over to you, Alicia, and thank you very much for giving the keynote of this, your very own Mexpopgen5 edition. Thank you.
Thank you very much. Can everyone hear me? Just checking.
Yes, we can, Alicia.
Okay, great. Sorry, I don't turn on my video. The connection is not very good here. So today I'm going to talk about the microevolutionary processes that happen at the genetic level and how they are connected to the species diversity using as a study system the Mexican Sky Islands. The ideas I'm going to present here started several years ago when I was an undergrad at Daniel Piñero's lab, who you know we are celebrating later on today in Mexpopgen. In this lab, I did the phylogeography, which is what we basically were all doing at the time, of this very nice juniper, who happens to be a rare species because it is isolated in different populations that are very spread among them and that show a high level of genetic differentiation among them. Later on, we saw that there are at least 3 lineages for this species, which are diverged around millions of years ago. And as an undergrad, this was very surprising to me. And Daniel Piñero, seeing my curiosity, asked me to keep asking what else could we learn about evolution based on these findings. So for my PhD, which I did abroad, I thought about the following. First, fragmentation and isolation can generate rare species because, as I showed you in this juniper, each of the different populations were separated by millions of years and are on the way of being different species or even could be considered different species. And these species will be rare because they will be localized in fragmented locations. So rare species by definition are endemic. And this is where things become interesting because some regions of the world have higher number of endemic species than what can be predicted based on macroecological variables. And these regions are mostly tropical mountains or islands, and which I'm highlighting here in this map. So I started to wonder why tropical mountains are so diverse. There are different explanations. A very accepted one is that tropical mountains are both promoting biodiversity persistence and diversification. They promote biodiversity persistence because they are areas of slow climate change velocity. This means that the species can persist in relatively the same area without having to move long horizontal distances, tracking climate as climate changes with, for instance, the climate fluctuations of the Pleistocene. But instead, they can remain in relatively the same area, just going up or down a slope. At the same time, mountains are great at promoting diversification because they create a set of heterogeneous habitats which are fragmented and therefore isolated. This has been used to explain why mountains are so rich in the species and in particular in endemic species, and has been tested using species distribution models and divergence times. However, I found that there was lacking a connection at the microevolutionary level linking how genetic differentiation among populations could lead to this diversity at the species level. So therefore, I decided to explain the origin distribution of biodiversity, looking at this link between the micro and the microevolutionary processes, and to test it. I use Mexican mountains as a study system because they are great, they are mountains, they are tropical, and they are in my beloved country. Specifically, I decided to focus in the Trans-Mexican Volcanic Belt, which is this region that you can see highlighted here in yellow, because it's an area that is relatively recent in origin, that is like if it were young mountains, but also old enough to have seen the different climate changes that Earth has gone through in recent history. Specifically, I decided to focus in the highest elevation volcans, which can be considered sky islands because they are these cold habitats. You can see me here wearing a hard jacket, immersed in relatively warming areas. This is Puebla City, and the other side is Mexico City, and I am sitting here in La Malinche looking at Popocatépetl and Ixtaccíhuatl volcanoes. So they are called sky islands. And islands, if you know a guy called Darwin or another guy called Wallace, they are great systems to study evolution because they allow us to have a system that is more or less isolated from the rest of the very complex processes that biology is immersed in. So to ask how to link micro and macroevolutionary processes in tropical islands, in the last few years, I have been asking the following questions. First, do populations of sky islands persist and differentiate in the same mountain through the Pleistocene glacial cycles as a function of topography? Second, does the colonization of alpine grasslands occur by dispersion from other highlands or from lower lands of the same mountain? And third, does the differentiation caused by topographic variables accumulate from the genetic to the species level? And these are the three questions I'm going to answer in this talk. So for the first question, let me explain it a bit better than what I did before. Imagine that you have a set of tropical mountains and a species like this flower that inhabits at a particular altitude where there is a temperature that it is adapted to. But this is an interglacial, like today when things are warmer. Then during the glacial periods, when global temperatures became lower, this species will be found at lower altitudes where the same temperature that is adapted to occurs now. Notice that this can mean that in some areas there could be connectivity between two populations that were previously isolated, and that between other mountains, depending on the shape of the landscape, this connectivity can not exist. If we think about this as cyclical, it means that populations will be going up and down a slope through history, and that some populations will go extinct or be ephemeral, like the ones here in the lowlands, whilst other in the mountains will remain through these glacial cycles. In a real setting, this is La Malinche volcano, or Sky Island, alpine grasslands are around this altitude, whilst during the glacial periods, they were around 1,000 meters below. And another important take-home message is that depending on the shape of the landscape, even if the geographic distance between is the same, it might be different effective distance depending on the shape of the landscape and whether two populations could be connected during the glacial periods or not. Okay, to test for this with real data, I sample all these mountain peaks looking for Juniperus monticola and Berberis alpina, which are two shrubs that grow at high elevation altitudes. And I develop a double-digest rat sequencing dataset. And we tested this in a specific, spatially explicit manner using both effective distances and effective distances considering both a flat landscape or the distribution of the species at different altitudes or with different models. For the first question is, do alpine grasslands persist in a single mountain during the glacial or interglacials? We found that yes, they do persist according to our climate modeling. You can see here in bluish colors how these grasslands occur in the same mountains both during the present interglacial and during the glacial periods. And also this is the same we found with pollen records. We tested this with genetic data using species, the allele site frequency spectrum, which is a method developed by Hugh and Hickerson, to test whether the species share a demographic history that coincides with the long-term population persistence. We found that indeed this is the case. We found that basically for all the populations of these two species, they have at the same time events of population expansion or stable population size. And this is congruent with our hypothesis of long-term population persistence in Sky Islands. That is, with populations surviving in the same mountains through the glacial cycles of the Pleistocene. The next part of the question is how does historic connectivity, which we have said is given by topography, can influence population genetic differentiation? And for this, we used just plain geographic distance, that is like assuming the landscape were flat, versus the distance considering the present distribution of the species according to a species distribution modeling or according to the altitude where they are known to occur. This will represent the interglacial periods. And then we also tested for the glacial distribution of the species, also using altitude rasters and species distribution models. We found for Berberis alpina that the glacial distribution better explains population differentiation, because this is a species that today occurs at about 4,000 meters above sea level. And despite that, the glacial distribution, which according to Poland records should be 1,000 meters below, better explains the genetic differentiation among their populations. We found the same for Juniperus monticola, because the one of the models of the climate of the glacial era better explains its genetic differentiation. So first question, the answer is yes. Populations persist and differentiate in the same mountain, and this is a function of topography. For the second question, I wanted to ask whether colonization occur from other mountaintops. This could be from long-distance colonization or through the glacial areas, as we have discussed before, followed by allopatric differentiation. Or alternatively, if colonization occurred from lower lands, followed by parapatric differentiation. To test this, Adriana Oscanga did his master thesis with Daniel Piñero and I. And she sampled these 7 sky islands in alpine grasslands and forests at different elevations. And she sequenced, with Sanger sequencing, each of the specimens we found of different Coleoptera and spider species. If we had the first option, that is allopatric differentiation, we'll be expecting Otus to be distributed in a single vegetation type, but in different mountains, while at the same time finding genetic differentiation among individuals of a single lotu in different mountains. On the other hand, we will expect, in the case of parapatric differentiation, to have Otus present in both vegetation types and phylogenetically close Otus to be present in each vegetation type. Now let's see what we found. First, I'm going to take a sip of water, sorry. Oh, delicious. Okay, first, Otus are mostly found in one ecosystem or another, as you can see in this figure, but rarely in both. We only found like this Otu and another one that and this one that were present in both. Therefore, this is more congruent with the hypothesis of allopatric differentiation. Then we also found that Otus are formed by individuals from a single vegetation type. For instance, all these are in orange, but from different mountains, like here in Cerro San Andres, Iztaccíhuatl, Toluca, Perote, among others. And again, this is congruent with the hypothesis of allopatric differentiation. We also found that there is genetic differentiation among individuals of the same Otu that inhabit different mountains, that this genetic differentiation corresponds to the distribution of each of the mountains. I'm highlighting here the results from one of the spiders, but we found similar results for 2 Coleoptera and spider families. So again, this is congruent with allopatric differentiation. So far we have a lot of evidence for allopatric differentiation, but we really wanted to test hard if there was parapatric differentiation happening. And this was the thesis of Cristian Quintero, which I did in collaboration with Dr. Juan Pablo Jaramillo. He sampled Lacaria trichodermophora, which is this beautiful fungus that grows in a gradient, in an altitudinal gradient in these volcanoes. So we sample in these 3 sky islands at low elevations with Pinus Montezuma forest and at high elevations within Pinus hartegi forests, which has considerably colder conditions. And here, Christian developed a GBS dataset to test these ideas genome-wide. So we found a lot of evidence for genetic structure separating different mountains. As you can see, this is Toluca, La Malinche, and Nevado de Colima, Toluca, sorry, which also is shown here in this tree and structure analysis. But when we tested for structure separating between the high and low elevation samples within the same mountain, we didn't find the structure genome-wide, and we didn't find either lossy under-selection. This doesn't mean that there aren't lossy under-selection at low and high altitudes. Probably there are, but probably these are few because we didn't capture them with this data set in a relatively small genome. And this means that there is no fingerprints of this structure genome-wide, while allopatrically speaking, in different mountains, there is this signature. So, in summary, there is higher allopatric than parapatric differentiation in the genome of Lacaria trichodermophora, which is a species that inhabits mountains at different altitudes. So, our results altogether show that whilst both of these options can happen and probably occur in nature, we found that colonization from other mountain tops followed by allopatric differentiation happens more frequently than parapatric differentiation from lower lands. The third question I want to ask, or we wanted to ask, is if differentiation caused by topographic variables accumulates from the genetic to the species levels. And to test for this, or before explaining you this, let me recap a bit of the neutral theory of biodiversity key points. The first is that dispersion plays a key role in distribution patterns. And this, I would like to remind you, it's a neutral process. There is also a spatio-temporal continuum of biodiversity that is reflected in a fractal geometry where we can see the same patterns in different hierarchical levels. That is, from the genetic level, then genealogies, species, genus, and so on, different hierarchical levels. If this were the case, if differentiation were accumulating from the genetic to the species levels in the tropical mountains, then we will expect to find a correlation between differentiation and distance at different hierarchical levels. This is something that has been tested before by Baselga and colleagues, but at very wide geographic distances across basically all of Europe with these very nice aquatic beetles, where they found that indeed the decay of similarity is the same at the haplotype level to different genealogy levels to a species level. Therefore, we have this fractal pattern of biodiversity decaying in similarity with distance. So we wanted to test this as part of Nancy Galvez' PhD thesis. I'm not going to provide a lot of details here because Nancy is also giving a talk in this Mexpopgen tomorrow. So for whole details, don't miss her talk. So what we wanted to test here is these processes that I just explained you, but it's a micro scale of within a single sky islands. Because if this were happening within populations on a single sky islands, which, as I told you, we have proved that biodiversity can persist there through time, then that will mean that the islands are generating diversity not only by differentiation species from one mountain to the other, but also by serving as a sort of source of new species within that single sky island. Another sip of water. Okay, so what we did in Nancy's work is that we put hundreds of pitfall traps in these different points that you can see here, distributed in well-conserved abies forests. And with these pitfall traps, Nancy made biodiversity soups of all the arthropods and other creatures that fell there. And we perform a DNA metabarcoding sequencing. And then, using a very new bioinformatic pipeline, we were able to distinguish from haplotypes to species and lineages at higher levels than species. What we found is that indeed similarity decays with distance in a fractal manner from haplotypes to higher levels. Haplotypes are shown here in blue, and higher levels are shown here. This is a clustering level, like higher than a species, and the species level will be around here, which is the greenish color. So as you can see, we found the same fractal pattern that I was telling you theoretically. And we also found that although this occurs in different taxa groups, for instance, in Diptera and Collembola, it is more marked in Collembola, which are these tiny arthropods that basically sit in the forest ground and live there without moving much, while Diptera are flies and things like that have wings and can fly, and therefore are suspected to be good dispersers. So we found that both processes occur in both species, in both groups, but that it's more marked in those that can disperse poorly. So, in short, to answer our third question, we have shown that indeed species and genetic diversity are linked because the differentiation that we have seen at the genetic level can accumulate in a fractal model to the species level. As a way of closing, I would like you to take home these following messages. First, tropical sky islands foment the long-term persistence of biodiversity, that is, that populations can persist within the same mountain during glacial and interglacial periods. Then allopatric differentiation plays a prominent role because as populations are isolated in these mountains, they keep differentiating from populations from other mountains, even if they can be in contact during the glacial periods. And finally, differentiation processes, which are driven by dispersion limitations, which are neutral processes, can accumulate from the haplotypes to the community level. And taking all together, I think that these 3 points help to explain how the sky islands of the Trans-Mexican column bars are cradles of biodiversity. That is, biodiversity is generating here, but also is persisting here. And this is happening from the genetic to the species level and helps to understand why tropical mountains are so diverse. With that, I will finish. Thank you very much, and I'm happy to take questions.
Great. Thank you very much, Alicia. That was a wonderful talk. So yeah, I encourage people to put your questions to the question tab on your right side of the panel. I think we have we have actually a couple of minutes for it, not many questions. So there's one question from Jorge Soberon. It says, great talk. Do you confirm Dan Jensen's hypothesis that mountains are taller in the tropics?
Yes, actually, that's something we discuss a lot in Nancy's paper, which I forgot to say is under review. Yes, tropical mountain passes are harder to cross in the tropics because the species are kind of like adapted to the temperature they live on. And because they can just move up or down the slope during glacial periods and remain in that temperature, they don't need to evolve adaptations to just cross the valleys and go to a different mountain, as it will happen in others. So yeah, our work supports that hypothesis, and I love it. Thank you, Jorge. Great.
So while we wait to have another question on the chat, I wanted to ask you somewhat a different question, Alicia, because I was wondering, this is great to think about the ecological structure of these mountains and really how species have differentiated and evolved in those islands. But besides species that you can sample from the ground and that kind of approach, have you thought, or do you think it could be possible to find differences in, for example, the microorganisms in the air? I mean, if you could take like air samples and compare between, whatever is, happening about, at 5,000 meters above sea level in the Iztaccíhuatl compared to Amalinche, if that could be different, maybe as a function of how polluted the area is because of being closer to Mexico City Valley or things like that. Can we kind of project those kind of models and say something about the air quality and the life in air?
Wow. I had never been asked that one. I thought you were going to ask me if people have done this in birds, and I was going to say, oh yeah, there's a lot of work in birds showing that either if they can fly between mountains, they actually don't do that a lot, and they are differentiated in the highlands. Well, I had never thought about sampling the microorganisms in the air. What we do know is that air pollution causes trees to be in trouble. This is part of the project that Veronica Reyes is going to present tomorrow also. And we also think that in these trees there is genetic diversity that is allowing some of them to better cope with pollution. But we're also looking at the microbes communities that live, specifically fungi, that live in their leaves. Because we think that maybe they could be playing a role. So they are not in the air, but they are in the leaves of the trees, which are in contact with the atmosphere. So yeah, looking forward to see those results. But this is still very in a preliminary stage.
Great. Yeah. Looking forward to see
that.
Okay,
I think we are finishing this session.
We are actually right on time. So if there are no more questions, I think we can move on to the next session. Thank you very much again, Alicia, for your wonderful talk. And yeah, again, congratulations for organizing this again.